Hello and welcome to Naked Genetics, the show which seeks to find out just what is going on in your genes. I'm Will Tingle, and join me as we seek to cover the latest in genetics research and see just what the front line of genetic mapping and sequencing can reveal about our world. In a particularly aquatic episode of Naked Genetics, we look at why a genetic bottleneck created decades ago means some whales are in deep water, how wastewater is helping genetic sequencers track down deadly diseases, and Surf's Up, how one beach-dwelling creature is shredding the gnar in order to find food. First up this week, let's look at a big story in the genetic sphere and look at some of the deeper questions being asked by geneticists. Let us link back up with my two genetics gurus, Shivani Shukla and Ailwyn Scally. Now, this month's genetic story concerns the genetic legacy of actions taken decades ago that are still causing issues today. The bowhead whale is the oldest living mammal in the world, with an estimated natural lifespan of 268 years. They live in and around the Arctic Ocean, feeding off krill, and, like many whales in the 19th and 20th century, had their numbers decimated by commercial whaling, from an estimated 50,000 individuals down to as low as 4,000. Since the whaling moratorium of 1986, numbers have recovered up to around 10,000, but now there are concerns that the lingering effects of intense whaling are rearing their ugly bow head. Now, I promise this wasn't even me that selected this story this time, so Ailwyn, if whaling has been, for the most part, banned since 1986, why are we now starting to see, or think we're starting to see, a potential reduction in their population?
They may be declining or going up or down for all kinds of reasons. One of the main factors that's thought to affect populations in the wild is climate change, and Arctic environments are very susceptible to climate change, more so than others. And the concern is about what might happen in the future rather than necessarily what's happened since whaling, since I think it's generally been a good thing for the whales that we've stopped hunting them.
And if that is the case, then, if climate change is driving such dramatic shifts in environment, you're going to need to be able to adapt pretty quickly, and that might be a problem for bowhead whales going forward.
Yeah, that's the thing, and that's the general kind of idea in conservation genetics. And simply put, that's how different are two individuals typically when taken from the species or from a population. And if they're very similar to each other, that means there's less variance or less versions of a given gene in the population. And if some big change comes along and the natural selection can only operate on versions that are already existing by and large, if there's nothing there, if there's no material for it to work on to adapt to that new environment or that change, well, then there's a danger that the population declines and perhaps even goes extinct.
And Shivani, given that this is in stark contrast to, say, a genetics study on humans, where we can resort to stuff like the UK Biobank and have huge swathes of data we can trawl through, how do you go about sequencing a population of wild whales?
First of all, you have to choose which whales you want to sequence. So in this case, they had the bowhead whale and the narwhal. These two species were great to sequence because they live in very similar habitats. And one kind of underwent a reduction in their population size because of a bottleneck event. In the Pliocene era, there was a supernova explosion. So their genetic diversity is fairly low right now, but that's for environmental causes. But on the other hand, the bow whales have low genetic diversity because of hunting. They have similar, essentially, diversity levels, but different reasons of why that happened. But they're wild, so that makes it difficult to track them down and sequence. So what they did was they collected tissue samples from Inuit hunters who have been hunting various species of whale for thousands of years. And they took these tissue samples between 1982 and as early as 2020. So you have the tissue sample, you can extract the DNA, it undergoes DNA sequencing, and you can compare heterogeneity between the DNA to work out genetic diversity in that animal.
So was there any striking difference in the genetic diversity across this timeline?
At present, the genetic diversity in both the species is fairly similar, but the main difference is whales live a very long time. So the narwhals live for 100 years, but the bow whale lives for 200 years. And that makes it very difficult for them to undergo genetic selection for favorable traits when each generation lives so long. And things are changing very quickly. The ice caps are melting, the temperature of their climates are changing rapidly. So that means bowhead whales are less adaptable because they're living essentially twice as long as narwhals. So even though currently the genetic diversity are both quite low, bowhead whales are in more danger, essentially. So I think that's helpful for people who are kind of looking into conservation, because it gives us an idea of which species need protection and why that's happening. Clearly, for the case of the bowhead whales, it's because of this whaling that's been happening in the last few decades.
Given that we stopped whaling in 1986, that's less than 2 generations of bowhead whale that have managed to be produced since then. It sounds like they're really in quite bad shape, aren't they, Ailwen?
They certainly are, by the looks of it, in trouble. I don't know if they're formally classified as endangered, actually. That's something...
Well, this is an interesting thing because the whole of bowhead whale, the group of them, because there's 4 different species, in total, on average, they're least concern. But yeah. I'm really concerned looking at this.
Well, it's worth bearing in mind that the actual risk for a species of extinction comes from absolute low numbers. So low numbers of individuals. So there are species that can survive for a very long time and exist with relatively low genetic diversity, and there are others that have much higher genetic diversity. And a good example of that is also one very familiar to us, humans. Humans also have gone through a bottleneck in the past, say about 50,000 years ago. And as a result, our genetic diversity is relatively low compared to the reality that there are billions of us. We might be worried about human extinction, but not for the kind of reasons that we're worried about with the bowhead whale. So although humans aren't as low in diversity as the bowhead, nevertheless, it's not a direct risk in itself, just having low diversity. They're not going to sort of go inbreed themselves to extinction. If we stop, if we continue to stop whaling, and if we manage the population and try to protect them from the risks that they face, then they should recover their genetic diversity and the future should be bright for them. And that's the case for bowhead whales and other endangered species like mountain gorillas, which also have extremely small numbers and extremely low genetic diversity, and others all around the world.
Shivani raises an excellent point as well, in that a study like this is super useful because it puts another perspective on what we need to conserve. Because if you just took it by raw numbers, you'd want to conserve certain things. But if you take it on risk of genetic diversity collapse, you would have to factor in another thing. And maybe you'd realise that there are certain species out there that need more conservation than others.
Yeah, well, that's also where the politics do come in, because it's not always clear what you should be working to conserve and what are the natural sort of units of conservation in a given species. Do you try to make sure that the group as a whole is preserved, or do you focus within that on subgroups that have their own unique identity, and then how do you identify those? And so some of this particular study was devoted to actually within the narwhal in particular, saying, well, actually maybe there are subgroups of the narwhal that have their own identity, their own genetic identity, and maybe we should designate them as separate units. And sometimes when that happens in a species or with a population, that means that there are new resources made available to do that, and perhaps even more money, although there isn't usually a lot of money going around for these kind of things. So as a result, these things can become very political. And I remember being involved in a study one time where the conservationists were very keen to identify a new species of orangutan, because that would change its status within the conservation system and within the government system in Indonesia, and therefore enable them to do a whole bunch of things that they wouldn't previously have been able to do. So there are a lot of different factors that are at play when you're trying to identify or trying to describe the genetic diversity of these species.
Thank you very much, as usual, to Shivani and Ailwen.
Music in the programme is sponsored by Epidemic Sound, perfect music for audio and video productions.
You're listening to Naked Genetics, the show about genes and the geniuses that study them. And now it's time to link back up with my new best friends at Illumina to look at how genetic sequencing is revealing more about our world. Today, I'm taking a step outside the office to get some fresh air. It's a lovely spring day here, and I need no excuse to take a walk after the winter we've just had. Rubbish weather, and of course, various winter illnesses. No one likes being ill, and even some of the most ubiquitous illnesses can pack a punch. It's not uncommon or particularly unreasonable to seek out a doctor and get a course of antibiotics. But, as I'm sure you well know, antimicrobial resistance, when a bacteria, fungi, virus or what have you becomes immune to classical treatments, is fast becoming a point of serious discussion.
I'm always thinking about the antimicrobial resistance personally. The latest estimates put that close to 5 million deaths annually, attributed to drug-resistant infections, and that's projected to skyrocket over the next 30 years if we don't do anything about it.
That's Illumina's Courtney Gonzalez painting a pretty grim picture. So, how does Illumina's genetic sequencing come into this? And why am I walking outside for seemingly no reason? Well, on this genetics programme, it's time to talk about wastewater. That's why I'm outside one of Cambridge's wastewater treatment plants, to really soak up the atmosphere.
So wastewater is everything that goes into your toilet and in through the sewage system and to the sewage treatment plants where the material is processed and cleaned up so that infectious diseases and other nasties don't get out into the environment. We looked at a variety of different sources of wastewater. So that can be things like a manhole in a city someplace. It could also be the wastewater coming directly out of, say, a dormitory at a university campus or a building of some sort. So all of those different sources will have sort of like different concentrations of material. If it's a manhole cover that's servicing, let's say, 50 to 100,000 people, then it's going to be a very different composition than something coming straight out of a building.
That's Illumina's Scott Kirsten, and I'm now very aware that I'm standing near other people's waste, so I'm going back inside. That's better. So, why sift through wastewater? What secrets could it possibly hold that would be worth all that?
Generally, wastewater surveillance, I think, is done for three main reasons: to monitor, track, and screen. So yeah, you want to monitor for evidence of infection within a community, track trends in those infections within the community, and screen for infections that could trigger additional testing or mitigation measures.
So my work in wastewater started out at the beginning of the pandemic for the detecting of COVID. It was a very convenient way to be able to non-invasively test a large number of people and track the presence or absence of COVID in samples and what kind of strains that are in there.
Since you do end up passing a lot of the microbes in your gut out when you do your business, monitoring wastewater is a passive but very insightful cross-section of the disease-causing microbes found in a population. And amongst these nasty bugs are the aforementioned resistant ones.
Agencies like the CDC and the World Health Organization issue these priority lists of microorganisms with drug resistant that are particularly threatening to human health. So things like methicillin-resistant Staph aureus or MRSA. So since we're not actually isolating and growing up these pathogens in a lab, we're just looking at the genetic sequences, we have to rely on the markers of drug resistance, so these antimicrobial resistance genes and variants, and at the same time, evidence of the bacteria that can carry those markers.
So what was limiting about previous methods of sequencing when it came to tracking antimicrobial resistant species?
So, on one hand, you kind of have PCR, which can enrich and pull out signal from these low concentrations, but you have to have an idea up front what you're looking for, and it's maybe going to be limited to 10 or 20 things, in a typical multiplex PCR. And then on the other hand, you have shotgun metagenomic sequencing, which can, sequence everything in the sample. So it's hypothesis free, which is good for surveillance, but you're potentially going to miss a lot because it's at that really low concentration.
Multiplex PCR, where you amplify a select few sequences of DNA, isn't all that helpful because wastewater surveillance requires you to cast a very wide microbial net, and shotgun sequencing has a broader range of genetic material to identify, but often wastewater concentrations were too low. The old options are either a mile wide or an inch deep. But maybe a sweet spot could be found.
In the lab with the nucleic acids, we convert those nucleic acids into libraries, as we call them, DNA and or RNA libraries that will then go on to an Illumina sequencer and be sequenced. There are various ways that you can manipulate those library preps to benefit certain types of organisms. So, for example, we do hybrid capture enrichment assays a lot that fish out certain sequences out of the material and enrich for them. So then we don't have to sequence nearly so many, and we can sequence a lot more samples out of that.
So what we found in this study is that we have this sort of sweet spot of precision metagenomic sequencing, which includes this step where probes go in and hybrid capture targets of interest for hundreds of pathogens and thousands of AMR genes. So you get that sensitivity for low concentration targets, but you also get the breadth that you need to really see what's in the sample without knowing exactly what you're looking for upfront.
And by doing that kind of enrichment, you can also process far more samples than you could in the past for a lot cheaper cost. And that's really key for this field.
So a cheaper, more precise means of tracking antimicrobial resistance in populations, and the lower cost means it can be deployed more often, which gives you greater resolution of data across, say, a year-long time frame. Indeed, it has already spotted times in which resistance to prescription drugs was rising in certain populations.
Yes, actually, in the pandemic, there was a lot of speculation early on that drug prescribing practices were, nobody knew exactly what sick patients were coming in with, so they would be prescribed drugs just based on having a cold. And it turned out to be COVID, which an antibiotic would not be the right thing to prescribe for that. And so you were seeing more resistance in the wastewater as a result of those drug prescribing practices.
So with an eagle eye being placed on the particularly harmful microorganisms, what can actually be done with this data to reduce them as a problem?
So one thing we can do with AMR surveillance data in general is use it to inform empirical treatment decisions. For example, in the absence of surveillance data, physicians are going to rely on their own experience of what drugs do and don't work for, say, a given bacterial infection. But if you know from surveillance data that 90% of those bacteria in your community are resistant to the first-line drug, then you can potentially use a different drug and save lives. And especially in countries without a robust clinical surveillance network, I think wastewater surveillance has a lot of promise to fill that gap.
Yeah, and just to add to that, at least for the infectious disease type fields, monitoring wastewater gives you a little bit of a head start. They basically say that when you start seeing, say, COVID in a wastewater sample, then it might mean that in 3 or 4 days, then patients are going to start showing up in the hospital with certain symptoms. So if you can get ahead of the curve a little bit and understand if some new infection is popping up in the population, then you can better inform hospitals about what to expect as far as symptoms or how to treat the patients.
So it's an early warning system, and it shows you what drugs might be more effective. Is there anything this can't do? So it's an early warning system, and it can show you what drugs might be more effective. Is there anything this can't do?
Cure the disease?
Well, I suppose you can't have everything, but a fascinating insight into the world of sequencing either way. Thanks very much to Illumina's Courtney Gonzalez and Scott Kirsten. That certainly wasn't a waste. And stick around to the end of the show, as the gauntlet has been laid for a very special challenge between Illumina and myself. But to round off this episode of Naked Genetics, it's time to pack your travelling bag again as we explore another quirk of evolution. It's a beautiful day here on the Atlantic shores of South Africa. Much better than that sewage plant, in any case. The nutrient-rich waters around here play host to some of the most striking and impressive organisms on our planet. Great white sharks, humpback whales, seals, penguins, I could go on. All of that life and all of those natural processes will inevitably lead to a lot of waste. Not that kind of waste, we're talking about something else this time. The dramatic predator-prey interactions in these waters will inevitably produce detritus, half-eaten fish, even the odd whale carcass. With the right tidal forces, a very real beach like this one that I'm definitely on can become something of a buffet for scavengers. But in the world of scavenging, it's very much first come, first served, which is a problem if, for instance, you happen to be a snail. Now, obviously we're not talking about your run-of-the-mill garden snail that causes great heartache for my mum. The snail in question is Bullia digitalis, often known as the plough snail. It has another name too, but we'll get to that. They're a pretty common sight on the South African coast, growing to about 6 centimetres long. If you'd never seen one before, you'd be forgiven for thinking a shell had found its way on top of a flat jellyfish. The point is, this snail is not particularly quick. Indeed, during low tide, the snail's locomotion looks very similar to if you tried to swim breaststroke whilst tied up in a bin bag. Such is life when you only have one very wide foot. However, when the tides turn, so do the tables. Boulia digitalis is a special snail. It took its singular foot and saw not crisis, but opportunity, because this snail is a surfing snail. If Boulia digitalis senses a delicious meal in the same direction that the tide is flowing, it rolls onto its side, hoists its foot up like a sail in alignment with the waves, and surfs along the seafloor towards its delicious meal. Now, it's not quite smacking the lip of a quality nug, which are surfing terms that I'm definitely cool enough to understand, and sure, it can only hang one, but it's still very impressive. The only speed limit is the speed of the waves. This thing can fly past the scavenging competition with, crucially, very little energy expenditure. Once this gnarly gastropod has reached its dining spot, it uses a proboscis, because this snail is weird enough that I only now am getting to the bit where it has a straw, sticks it into the carrion and sucks up the soft tissue. Delicious. And, after all that, the other advantage of having one massive sail foot, when the tide starts to go out, Boulia digitalis can just plant the foot in the sand and stop itself from being pulled out to sea. The fact that a snail the size of a drinks can has the wherewithal to understand that tidal forces can carry it to food is remarkable, and long may this creature continue to shred the gnar. That's it for this month's edition of Naked Genetics, but before we go, do make sure to tune in next month for the start of a very special series of challenges. I've been hearing all about the impressive things that Illumina can sequence, and I want to go myself. So, Will It Sequence continues, with me finding out if I can fool the cutting edge of sequencing technology. Can Illumina work out what food I've been eating based on one swab of the bacteria that grows on it, and what actually does grow on your food? Find out next time. Well worth a listen. Until then, if you'd like to drop me a line, my email address is wilty@nakedscientists.com. I've been Will Tingle. Thanks for listening, take it easy, and until next time, goodbye.